Всім добрий вечір. По ідеї, ми зараз йдемо лайв стрім на Ютубі, а також дякую всім, хто приєднався особисто. У вас є привілегія запитувати Катерину все напряму. Отже, я рада вітати нашу запрошену лекторку Катерину Макову. Катерина наразі є професором в Penn State університеті. Вона почала свою кар'єру в біології, якщо не помиляюся, з Київського національного інституту. І потім отримала звання доктора та мала свій постдок в університеті Техасу та Чикаго. І, отже, наразі Катерина веде лабораторію, в якій вивчаються мутаційні профілі в мітохондріальній ДНК, в статевих хромосомах, зокрема хромосоми Y, та спеціалізується з питань неканонічних структур ДНК і ролі всього цього в еволюції та біомедицинських питаннях. I will switch to English now. Just to introduce everything for our English-speaking speakers. Thank you all for joining. And I'm sure if you found us, then you already know Kateryna, as I think Kateryna had the most to reach out to the English-speaking audience. And so please enjoy the lecture, and you will be able to ask the questions at the end when you unmute yourself. Or otherwise, you can paste them in the chat, and I will read them out loud. So, Kateryna, please, the microphone is yours.
All right. Thank you so much, Valeria, for inviting me. And I will be giving this presentation in English because my Ukrainian is a bit rusty. I moved to the US in 1995. So long time ago, but I will be happy to answer any questions you have either in English or in Ukrainian after my presentation. So today I'm going to talk about a work that is yet unpublished, but we have a preprint on bioRxiv, and in fact it was distributed, the link to the preprint was distributed with the advertisement of my talk. And it is a collaboration between myself and a postdoc in the lab, Karol Paul, who did actually most of the work, and I'm actually borrowing some of his slides. But also this is a larger team of collaborators, and I will introduce everyone as I go. So today I will be presenting on the topic of how and why multicopy genes survive on the human Y chromosome. And before I go into the detail of this question, I wanted to talk about the sex chromosomes in the human genome, the X and the Y. The human X and the human Y have now been deciphered in their entirety. We know every single base of these chromosomes. And human X is quite a large chromosome. It's 154 megabases long and has about 800 protein coding genes. At the same time, the human Y is much smaller. It's only 63 megabases long and has about only 100 protein coding genes. And the human females have two X's and human males have X and Y. But the question is, why are these chromosomes so different? Why is Y so tiny and gene poor, while the X is quite large and gene rich? So to really be able to address this question, we need to go back to the sex chromosome evolution in mammals. And going back about 170 million years, the sex chromosomes, our sex chromosomes X and Y, evolved from an ordinary pair of autosomes. And one of the genes, SOX3, on this pair of autosomes, acquired a mutation that was beneficial to males. And this gene became SRY, which is the male sex determining gene in mammals. And to really keep this male-specific mutation intact, the Y chromosome underwent a number of inversions. You can see that this part is flipped compared to the X. And in the areas of inversions, the Y chromosome could not recombine with the X chromosome anymore. And also in these areas of inversions, the Y chromosome started accumulating additional genes and mutations that are beneficial to males. But the X still kept the genes that are expressed and important both for males and females. Because of these inversions and inability to recombine between the X and the Y chromosome accumulated many deleterious mutations, also acquired some genes that moved from the autosomes, and underwent a number of deletions and degradation. As a result, it shrank in size considerably. So now it is a much shorter chromosome as compared to the X, because the X can still recombine with its other copy in females. So now the X and the Y can recombine over just very two short regions. They're called the pseudoautosomal regions. I'm showing the longer one here, but there is also another pseudoautosomal region here on the tips, on the other tips of X and Y. So summarize, the bottom line of the sex chromosome evolution in mammals is that the Y chromosome cannot recombine any longer over most of its length and is constantly bombarded by mutations that are mostly deleterious. Many of the mutations on the Y are deleterious, and there is no easy way for the Y to get rid of these mutations because it doesn't recombine with the X. So we were puzzled by these unusual features of the Y chromosome, and we studied this chromosome in humans and other great apes. But before I describe our work, I would like to introduce to you the structure of the human Y chromosome. And the human Y chromosome contains two pseudoautosomal regions. We call them pars, here shown in green. And also it has X-degenerate and ampliconic regions, as well as lots of heterochromatin. So the X-degenerate regions here shown in yellow, they have homologs on the X chromosome. That's why they are called X-degenerate. But the ampliconic regions here shown in this periwinkle color, they are unique to the Y chromosome, and they are highly repetitive. These are the regions that amplified on the Y. This is why they're called ampliconic regions. And within these ampliconic regions, the Y chromosome has many palindromes. So you can see P1, P2, all the way to P8. So what are palindromes? These are inverted repeats. And here there is a toy example of a palindrome, GACA. And then there is also GACA, but on the other strand. And then there is this interruption, which frequently has this loop structure, right? So the ability of palindromes to pair between the two arms on the same strand allows for the genetic exchange between the two arms and for gene conversion. And this is something that I will come back to in a minute. But for now, please remember that there are many palindromes, these inverted repeats on the Y chromosome and its ampliconic regions. So the Y chromosome has genes that are present in multiple copies. And these genes form gene families. Here they are represented here. There are a total of 7 gene families. Previously, we thought there were 9, but then later investigation identified that some of these gene families have only pseudogenes, so they are inactive families. And these gene families have copies that are highly similar to each other. By highly similar, I mean 99% identical in sequence, very highly similar. These genes and gene families function in spermatogenesis. You can see that many of them are involved in meiosis, in germ cell development, in fertilization, or germ cell apoptosis. So they really important for male-specific reproduction. And they are organized in these palindromes. So this is a cartoon of the palindromes, and there's another inverted repeat here as well, on the human Y chromosome. And these are the different gene families. And except for one gene family, all other gene families are organized in palindromes. And the single gene family in human that is not organized in the palindrome is TSPY, but this gene family is organized as a tandem repeated array. So the Y chromosome has many multicopy genes. In fact, out of 106 protein coding genes, 68 are members of multicopy gene families. And our goal is to study the evolution in great apes. So in fact, we're trying to solve the puzzle, how and why these genes survive on the non-recombining Y chromosome. Because the Y chromosome was suggested to have a fate of very quick death, right? However, this is not something that we observe. Still half of the humanity has the Y chromosome, and it's not going anywhere anytime soon. So by addressing the question of how and why the multicopy genes survive on the Y chromosome, we're also trying to answer a question why this chromosome survives as a chromosome as well. So very recently, we were part of several landmark studies. In 2022, the human genome was sequenced completely. 8% of the sequencing data were added to the previous version of the human genome. I believe that this is really a very important revolutionary discovery. However, one of the chromosomes was missing from this paper that was published in Science, and that was the Y chromosome. So in fact, the correct title for this paper should be The Complete Sequence of a Human Female Genome. So when this paper came out, I became very excited, and I approached the consortium, the telomere to telomere consortium. And by telomere to telomere, we mean that all the chromosomes were determined in their entirety from one end to another end. So I approached the T to T consortium asking about the Y chromosome. And they told me that the Y chromosome was being sequenced, and asked whether I would like to join forces in sequencing and analysis of this chromosome. So this is exactly what we did. And we published the sequence of the human Y chromosome in 2023. And this chromosome is very repetitive. So it has many palindromes, as I mentioned already. Also, it has a large heterochromatic region. So sequencing this chromosome and assembling it, putting it together, was definitely not easy. But using this approach, we later were able to sequence and assemble the ape sex chromosomes. So the Y and the X chromosomes from great apes. This is a study that was published in 2024. And more recently, just last year, we sequenced completely from telomere to telomere the genomes of great apes and one lesser ape. So we even got a cover of Nature magazine for this discovery. So what was done in this work in terms of the copy number determination for the sex chromosomes, in this particular case, the Y chromosome. So these are the gene families, the multicopy gene families on the Y chromosome. These are the species that we studied, and the circles and the numbers correspond to the number of copies of each gene family in each of the species. So before I go into these results, I would like to highlight the species that we used for this work. So this is human, and these are our two closest relatives, bonobo and chimpanzee. So the human lineage diverged from the bonobo and chimpanzee lineage about 7 million years ago. But the bonobo and chimpanzee lineages diverged from each other 2.5 million years ago. The next species on this tree is gorilla. Human and gorilla lineages diverged about 9 million years ago. Then we also have two species of orangutans, Bornean orangutan and Sumatran orangutan. And they separated from the human lineage about 17 million years ago. And these are the two most closely related species in our study. They separated from each other about 1 million years ago. And then we also have Siamang, which is an outgroup to the great apes here on the tree. And the Siamang lineage, this is a gibbon, separated from the human lineage about 20 million years ago. So now let's take a look at the numbers here. And there is a lot of variation. There are some gene families that are highly copious, have many copies like TSPY here. Also a lot of variation in the copy number among different species. And then there are some gene families that have fewer copies like VCY or BPY2, for example. Also, please keep in mind that here for this tree, we only analyzed one individual per species. There is also a lot of variation that we now observe among individuals for each species. But this is the data that we had available to us coming into the study that I will be describing to you now. And I would like to represent the gene conversion process to you. This is something that maybe you heard about before, and this is a genetics concept. And this is a process of unequal genetic exchange. So I will show an example of intrachromatid gene conversion. So this is the palindrome. So these are the inverted repeats. So one arm is yellow, another arm is blue. And because these are inverted repeats, they can recombine with each other, they can align, and they can recombine with each other. And if the genetic exchange is uneven, then there will be more yellow parts at the end of this process than blue parts. And you can imagine, for example, that there was a deleterious mutation in a blue arm, but then this deleterious mutation got replaced by just a functional site over the yellow part in a gene. As a result, the gene was saved, it was salvaged, but also the blue gene became more similar to the yellow gene, right? And this process is quite frequent. And we believe that this process is the reason why the multicopy genes on the Y chromosome are so similar to each other within each gene family. But also, this is the reason why the Y chromosome is using this intrachromosomal recombination to be able to save its genes. It's the survival mechanism for the Y. And in fact, we know that this process is highly efficient in palindromes. On the other hand, this process also appears to be working in tandem repeats. So if you have the tandemly repeated genes, they can also undergo gene conversion. So we knew that this is happening, but we definitely did not know the details. And what I would like to talk about today are 3 questions that we addressed. The first one is whether palindromes or tandem repeats are more efficient in homogenizing ampliconic gene copies on the Y chromosome. Then we also looked at the expression of ampliconic genes. And by ampliconic, I mean the multicopy genes, right? What drives the expression of ampliconic genes? What are the reasons behind the transcript diversity of these genes? Is this alternative splicing or is it copy number variation? And then we went even further and we predicted the protein structures of these genes. And we asked the question of how they were maintained. So again, I would like to acknowledge Carl. This actually is a project that he has been working on for the past 2 years. It's not a low-hanging fruit. It took a lot of work, a lot of very careful curation of every gene copy to really be able to obtain the results that I'm sharing with you now. So we're going to move on and we're going to address the first question about whether palindromes or tandem repeats are more efficient in homogenizing ampliconic gene copies. So how does this actually work? So we have these palindromes here, we have these inverted repeats, and you can imagine that these inverted repeats can align to each other, right? The arms of the palindrome can align to each other and they can exchange genetic information exactly as what I was showing on the figure to the right. But how does this work for tandemly repeated arrays, especially because you don't have the second chromosome to work with, right? So we propose this mechanism according to which the genetic exchange and gene conversion can happen between sister chromatids of the Y chromosome. But then the different copies of tandemly repeated arrays, which can misalign, but because they're so similar to each other, right, they can still exchange information and undergo the same gene conversion as for palindromes. The question is, which mechanism works better? Is it the organization with palindromes that leads to higher levels of gene conversion, or is it tandemly repeated array organization that leads to higher levels of gene conversion? So that's something that we did not know and wanted to find out during this study. So first we asked the question is how often the gene copies are organized in tandem arrays versus in palindromes. And we thought that mostly this should be palindromes. This is such an important feature of the Y chromosome in great apes. So what I'm showing here are the species that we studied and the different gene families. And for each of the cells in this table, there are 4 numbers. The first number is the number of copies organized in palindromes. The second one is the number of copies organized in arrays. And here in the parentheses, the third number, sometimes the copies are organized in both palindromes and arrays. Such cases are rare, but they do exist. And then sometimes we have copies that are outside of arrays or palindromes. So each gene family is different, but overall, if we look at the overall number of copies across all these families and all great apes, we have 163 copies organized in palindromes, 140 organized in arrays. So a very comparable number of copies exist in these 2 types of organizations. And we have only 46 copies outside of arrays or palindromes. So there is definitely this tendency of gene copies on the Y chromosome to be organized in either palindromes or arrays. And there is no big preference towards one type of organization versus the other. And then we directly address the question of which type of organization is more efficient in gene conversion. And the result of gene conversion is homogenization of sequences. In other words, the result of gene conversion is making gene sequences very similar to each other. So we looked at the sequence identity for genes that are organized in tandem arrays for the same array, for tandem repeats with different arrays, or palindromes on the opposite arms of the same palindrome. And what we found was quite striking. So the sequence identity of genes organized in tandem repeats was very similar to the sequence identity of the genes that are organized in the palindromes on the opposite arms of the same palindrome. But if we were looking at the genes that were located in the same arm of palindrome or in different palindromes or outside of palindromes or tandem arrays, then sequence identity was significantly lower. Therefore, we can conclude that both tandem arrays and palindromes are equally efficient in homogenizing genes via gene conversion. So this appears to be two alternative structures that work equally well in preserving genes on the Y chromosome. Another question that we had is whether the distance between the genes within the tandem array can determine the level of homogenization within the tandem array. And here, what we did is we plotted the distance between gene copies versus the sequence identity. And we indeed found the significant negative relationship, meaning that the genes that are located closer to each other have a high chance of undergoing gene conversion and homogenization as compared to genes that are located further away from each other. So I am about to finish this first part of the presentation, and we asked the question whether palindromes or tandem repeats were more efficient in homogenizing ampliconic gene copies, and the answer is that both. So gene copies are equally distributed between tandem repeated arrays and palindromes, and both structures appear to be equally efficient in homogenization. So I'm going to move forward and address the second question, and the question is, what drives ampliconic gene transcript diversity? And alternative splicing or copy number variation. So this was the question that we had coming into this study. So we were really fortunate because we had this really good data set of testis samples for all of these great apes. That's something that my lab has been collecting for over 20 years by collaborating with different zoos and other animal research organizations. So from these samples, which we had frozen in our deep freezers, we performed 3 types of experiments. First type of experiment was targeted long read sequencing of transcripts. So we designed probes to which we hybridized the cDNA synthesized from this total RNA. And these probes, they corresponded to the exons of the Y chromosome genes of interest. We also did untargeted long read sequencing of the cDNA from testis samples, and we also did short read RNA sequencing. So these were the 3 data sets that we had at our disposal, and they were mostly published previously. Then we collaborated with Alexandra Greshnova and Stefan Kanzar on assembling this transcripts with string type. So this approach allows one to use the reads obtained from the RNA through the cDNA synthesis and assemble the transcriptome and then compare the transcripts in terms of alternative splicing. So I'm going to show some of the results here. So these are 6 of the 7 gene families that we studied: BPY2, CDY, HSFY, RBNY, TSPY, and VCY. And you can see that there are several colors and several rows for each gene. We can take a look at the first gene, BPY2, and it has a total of 6 isoforms that we observed across great apes, and these are structural isoforms. So each colored block is an exon, and the introns are these lines between exons, and they are not shown to scale, while the exons are shown to scale. And you can see that there are quite intricate patterns of alternative splicing that are observed for the isoforms for this particular gene. And each of the structural isoforms is shown in different color. So by structural isoform, I mean different composition of exons within particular transcripts. So CDY gene has fewer isoforms, HSFY also fewer, RBNY has many different isoforms, TSPY has many again, and VCY has only a couple of isoforms. So this is just talking about the structure of transcripts in terms of exon presence, absence. Next, Carl analyzed the sequences of isoforms, because in addition to the structure, there is variation in the sequences of these isoforms, which comes from different copies of the genes. So Carl was able to decipher this variation as well. So what we have at the end is the collection of transcripts that depicts both sequence and structural isoform variation. So I'm going to show this with an example of just one gene, TSPY gene in Bornean orangutan. So here the colors correspond to the different isoforms on this plot. So in this case, this is isoform 37 and isoform 32. But the different bars are unique sequence isoforms, right? So we have a total of 1, 2, 3, 4, 5, 6 different sequence patterns. And the unique isoforms will be a combination of sequence and structure. So we have 6 plus 4, a total of 10 different unique isoforms here. And this is just one gene family and one species, but we did this for all of them. And the patterns are quite different among different gene families. So for some gene families, it's mostly the sequence isoforms that are present, not that much structural isoform diversity. But for example, for CDY. For some other genes, RBNY, for example, there is a lot of variation in structure, but perhaps less variation in sequence. So there is a lot of diversity in isoforms, both at the sequence and the structural level. One of the genes that we were interested in particular is TSPY. And TSPY displays this one dominant isoform, which is shown here in orange, and has just some minor sequencing isoforms present, minor other structural isoforms present, while it does have a substantial amount of sequencing isoforms because it has a very high copy number. So we also wanted to look at large-scale patterns in this data, and we asked the question whether the number of gene copies can predict the number of sequence isoforms. And the hypothesis was that if there are more gene copies, that there will be more sequence isoforms. And this is exactly what we observed. So on the x-axis, we have the number of gene copies, and on the y-axis is the number of sequence isoforms for each gene family in each species. And we do observe a positive relationship between the two. We next asked question whether there is a relationship between the number of exons and the number of structural isoforms. You can imagine that if a gene has many exons, then there are many different ways in which they could be included or excluded from transcripts, generating a lot of structural diversity. So this is exactly what we observed. We plotted the number of exons in the gene versus the number of structural isoforms. And again, we observed a positive relationship between the two. So to summarize this part of my presentation, we were trying to address the question, what drives ampliconic gene transcript diversity, alternative splicing, or copy number variation? And as a result, it's both. So particularly high copy number genes exhibit great sequence transcript diversity. On the other hand, genes with high exon counts produce many structural isoforms. So this was work done at the level of mRNA transcripts, but these are protein coding genes. So what is happening at the level of proteins? So what are the protein structures of ampliconic genes and how are they maintained? So we did not have any protein data, but we could predict protein structures. And you probably heard of AlphaFold. So we used an analogous tool called CollabFold. And this is a work that was done in collaboration with our colleagues from Seoul National University, Martin Steiniger and Sukwan Park. And what they did is they predicted the structures of all of these gene families of each transcript, in fact, that we identified from our transcriptome work. And here I'm showing the structures of five proteins that originated from these gene families. And you can see that they have the structured parts, right? Either alpha helices or beta sheets, which frequently are very important for their function. And two of the gene families encoded unstructured proteins. So they would look like this, right? Like this parts of the genes that are unstructured. So mostly I will be talking about the structured proteins for the rest of my presentation. So the CollabFold works in many ways, the same as AlphaFold. The only difference is it uses a different alignment algorithm, making it much faster. And it can predict the score of structure stability along the protein molecule. So here, what we can see is that there are some parts of the protein that are more structured than the others with high confidence. So this is a CDY gene. And now we can align the structure predictions to our isoforms. And what we found is that here blue means a highly structured part of the protein. That the isoforms usually share these parts that later encode the highly structured parts of the protein. This is what is observed here for the CDY gene. And this is later also something that we could see for the other gene families we have analyzed, is that the conserved parts of transcripts, the transcripts have exons present in all of them, usually have this structured, highly structured parts shown in blue. So this is great. This was really interesting. So how do we put all of this together now? So we have this transcript isoforms. So I'm just going to show this with an example of one gene family, TSPY. We have these transcript isoforms, and the colors correspond to the structural isoforms I was showing in one of the previous slides. But the many different rows correspond to the sequence isoforms. And then they can translate into protein isoforms. We usually have fewer protein isoforms than we have transcript isoforms because of synonymous mutations, right? If there are some transcripts that have synonymous mutations, they will still lead to the same amino acid. That's why we have fewer protein isoforms than transcript isoforms. But then these protein isoforms can be clustered based not on sequence, but on structure. And the striking observation here is that despite having many protein isoforms at the sequence level, there are just several, usually just one dominant protein structure cluster that encompasses most of the protein isoforms that come either from the same structural transcript isoform or sometimes even from different transcript structural isoforms. And this is a pattern that we observe for TSPY, just one dominant protein cluster. For RBMY, again, one dominant cluster, and CDY. And these are our high copy genes on the Y chromosome. The pattern is a bit more intricate for DAS. This is a very complex gene, which we analyze separately. And BPY2, VCY, and HSFY, they just don't have this much transcript diversity to start with. And as a result, they have several protein clusters, but they are similar to each other. So what is driving this conservation in structure? Why do we observe only usually one dominant protein structural cluster? And to address this question, we analyzed selection occurring at the sequence level in these genes. And this was done in collaboration with Sergey Pon from Temple University. We were measuring the non-synonymous to synonymous rate ratio. And synonymous mutations are used as a neutral background. So if the gene is evolving neutrally, you expect the number of non-synonymous to synonymous mutations to be very similar if corrected by the number of synonymous and non-synonymous sites. But the DNDS ratio less than 1 indicates purifying selection, and the ratio greater than 1 indicates positive selection. So we found significant purifying or negative selection for CDY, for HSFY, for RBMY, and for the RNA binding domain of DAS. So there is purifying selection for genes that encode highly structured proteins. We also observe purifying selection for highly structured domains of the proteins. Here I'm showing this for DAS. So the TSPY gene is an exception from the rule. It is a structured protein, but it does not present evidence of purifying selection, and we are still trying to understand why this is the case. But for me to summarize, so what are the protein structures of ampliconic genes and how are they maintained? We have evidence of purifying selection acting on the highly ordered proteins or protein domains. So how do we put all of this together? So let me summarize my talk. The gene conversion maintains the high sequence identity among functional copies. And this works really well both within palindromes and arrays that undergo this homogenization for the functional gene copies. Both alternative splicing and gene duplication generate isoform diversity. And then it's selection that maintains conservation of protein structure and function, despite some sequence divergence. So what we have is we have this multi-level buffering system which enables genes to tolerate mutations, both sequence and structural variation on this non-recombining and highly mutable chromosome, the Y, while preserving the spermatogenic functions essential for male fertility. And what are your take-home messages? First of all, is why? Why are genes still alive on the Y chromosome? Well, it's because many of them fulfill important functions and undergo purifying selection. How are they maintained? Many of the genes are multicopy and are organized in palindromes or telomerase. And when they are organized in such a way, they can undergo gene conversion. And gene conversion can facilitate their homogenization, which removes deleterious mutations from these genes. And I would like to thank all of our collaborators and also acknowledge Hanna Palova and Byung-Jun Ko, who also contributed to this work. But mostly is Carol, who did, I would say, 99% of everything that I was talking about today. We have this project completed mostly because of his hard work and our amazing collaborators. Thank you very much for listening, and I'll be happy to answer any questions that you might have.
Yes, Valery, please unmute yourself.
I have no specific questions, just wanted to say that it was amazing presentation, very interesting topic, great cartoons and colors. It was pure pleasure for eyes just to see. And also I learned something new, that palindromes form this kind of loops, which allows to exchange genes. It was something really unexpected from today's lecture. So, and it was the very beginning. So it was great. Thank you.
Oh, great. Yeah, I'm glad.
And I have a small question, but it's rather silly one. So you said that you had a lot of samples of testes of different apes.
Yes.
And what about human material?
Yeah, so for human material, we could get some data from the databases. We could also get testis samples from human as well, from a company, right? So apparently you can get this samples from cadavers and use in research. Yeah. Mm-hmm.
So you mentioned T2T consortium. I believe that they sequenced cell line, which was like specifically cultivated. But in your research, you use like real life data because of all this diversity. You needed a lot more than just online.
Yeah, so this is a very good question. So to generate T2T genomes, really high quality genomes, you need to use long reads, long sequencing reads. And they work best if you have really high quality DNA, right? So for this, it's best to use cell lines. On the other hand, practically all the genes I was talking about today, they're not expressed in the cell lines that are used for T2T genomes, which are fibroblast cell lines or lymphoblastoid cell lines. They're only expressed in testes, right? So we had to use the real material to be able to study the expression of these genes. Mm-hmm. But then we could align them with the T2T genomes. Mm-hmm.
Thank you. And also, I remember that on the slide with gene families distribution, I remember that some data was missing. Like this gene family is missing in some species, and so surprisingly for humans, all like 6 families was present. Is this like just because you were focused on this, like gene families in human chromosome Y, or we just have the most of all the apes?
It's a very good question. So our goal was to trace human gene families, right? And some of these gene families got lost from the Y chromosomes of other great apes. On the other hand, there are some gene families on the Y that are present in some great apes, but are absent from the human Y chromosome.
Okay, thank you.
Sure. Any other questions?
Please, Sergey, you can unmute yourself.
Hello, Kateryna. Thank you so much for the talk. Very informative and very interesting talk. So I wanted to ask you an evolutionary question. So you described this classical situation when our species and apes also got entrapped into Muller's ratchet, right? So when more and more mutations accumulated on the Y chromosome, and then these compensatory mechanisms of kind of interchromosomal recombination emerged to stabilize this chromosome, right? And we know that some of the males in our population have XYY karyotype, right? So those are once in a thousand type of event. So do you think why this mechanism didn't develop as a default protection rather than the recombination facilitated by tandem repeats? Is it a future of our species, or is it not possible?
It's interesting, right? So it's rare, right? It's rare that there are XYY males. Sometimes there are even 3 Y chromosomes that might be present. In these cases, potentially the 2 Y chromosomes could recombine with each other. We don't know that for sure. But I think this would lead to disbalance, right, in dosage as compared to the X chromosome, and this is why this mechanism is not used that much, right? So I think the gene dosage might be disbalanced between X and Y for the genes that are homologous between the 2 sex chromosomes. So you would have 3 copies, right, in males, while 2 copies in females.
Yes, surprisingly, these males are doing quite well. So they are higher, they are fertile, and compared to other abnormalities, other chromosomal abnormalities, they're fine. But this cannot be inherited. This condition cannot be inherited. So their children still will be XY. But still, are there any genetic research about this subpopulation in apes, maybe, or among NBA players, or in just to understand this subpopulation, or is this just an unknown area?
I think it's unknown. I think it's very interesting. I completely agree with you. It would be nice to do more research on this, and we just don't have this data right now. Frequently, this condition is also arising somatically, right? So not from the very beginning, these males are XYY, but during the development or even later in life, some particular cell lineages are acquiring this extra Y chromosome. So apparently it appears to be lethal at the fertilization or early embryonic stage.
Okay, thank you so much.
Yeah, sure.
Do you see something similar in apes as well, like XYY apes?
I honestly don't know. I have not seen this, but it doesn't mean it doesn't exist. I don't know.
Until there's other questions, if I may, I will jump in. So this is fantastic work, and congratulations on it and on your contribution to T2T. So what surprises me about these particular genes that you studied is that many of them have a lot of copies and functional copies, right? So the fraction of pseudogenes, as you showed, it concerned only 2 gene families. So I wonder why would, I know it's not a good question in biology, why something is needed for evolution, but still, why all of the copies are needed. So usually the copies are made for the redundancy, so the function is not lost in very important processes, but so many. And I wondered if it could be that some of them can be regulated in a different way, having different like promoters or enhancers that would give the same function but under different conditions.
Yeah. No, I think you are asking something really interesting. First of all, I agree that the main reason might be the genetic redundancy, right? So because the mutation rate on the Y chromosome is so high, and it makes sense to have many copies, right? Because if even some of them are bombarded by mutation, you still have others to work with, right? And you still have others that serve as the templates to rescue the copies that died out, right, or accumulated deleterious mutations. Yeah. But now the question of the number of copies, it should correlate with the levels of expression, right? It should also correlate with the amount of protein. But this is something for which we currently don't have the data for. But it would be really nice to be able to prove this, right? Looking at multiple individuals from the same species, maybe in humans. Yeah. Something that would be challenging to do with the data set that we currently have, but hopefully we can address this on a larger scale in the future. Mm-hmm.
That's interesting. Thank you very much.
Yeah. Any other questions?
Okay, it seems that none for now. But I recall that Kateryna, you've been at the USAB in 2025, right?
Mm-hmm. Yeah.
Do you know, like, if you would be implied in similar initiatives? It would be really lovely to see you again among the informatics community.
Absolutely. No, thank you so much. And I hope that everyone can have my contact information in case you have any questions about this work or you need any career advice. I'll be happy to address that. You can find me easily online. Yeah, but also through Valeria. Valeria has my email address. So yeah, just get in touch and I'll do whatever I can.
Thank you very much. And it would be lovely to see you on upcoming also international conferences on evolution or genomics. Thanks a lot for your time and great work.
Of course. Thank you so much for having me. Yeah, it's great to see everyone. Yeah. Okay. Goodbye.
Bye. Wish everyone a
Thank
good day.
you.
Bye.